series of previous lectures, we have considered the language, main concepts and methodology of the evolution, such as, for example, phylogenetic comparative analysis of genetic sequences. Equipped with these skills, we analyzed currently available information on COVID-19 in order to shed some light on the evolutionary origin of this human disease. In this session, we start our journey through human-specific evolutionary events, such as split of the genus Homo from the great apes, archaic hominin species, and ancient migrations shaping our genetic diversity and human-specific diseases. We are starting with the topic closely related to zoonosis of COVID-19, in particular, non-human primate origins of human infectious diseases, and then we will progress towards finding out more general taxonomic relationship with the extant great apes. Non-human primate parasites represent a substantial reservoir of potential human pathogens, which may be able to make the jump from wild non-human primates to humans because of the close phylogenetic relationship. The two major human infectious diseases affecting Africa are both of zoonotic origin from the other great ape species. First is the human immunodeficiency virus 1, HIV-1, causing AIDS from chimpanzees, and the Plasmodium falciparum protozoan, which causes severe malaria from gorillas. Let us watch a short SciShow video outlining the sequence of HIV zoonotic events.
All doctors knew was that suddenly, healthy young gay men were developing extremely rare infections and cancers, and it was killing them. One of the first people to notice the pattern was an immunologist at UCLA. Between the fall of 1980 and the following spring, he saw a string of 5 patients, all gay men in their 20s and 30s, with an unusual kind of pneumonia. There was a fungus growing inside their lungs. Normally, the fungus was totally harmless and would never infect the lungs, but in these men, it had, and it was making it hard for them to breathe. The patients also had oral thrush, basically yeast infections in their mouths, and very few CD4 T cells. By June, when the immunologist wrote up the results for the CDC's weekly mortality and morbidity report, 2 patients had died. A month later, a dermatologist in New York chimed in with a similarly disturbing report, this time with Caposi Sarcoma, a rare cancer where patients develop blotchy purple lesions on their skin. In 2.5 years, 26 young gay men in New York and LA had been diagnosed with Kaposi's. Some also had the weird fungal pneumonia, and 8 had died. It's hard to imagine now, but at this point, scientists had no idea what was making people sick. They didn't know if it was some sort of toxin or a pathogen, and if it was an infection of some kind, they didn't know how it was spreading. That meant they couldn't warn people about how to protect themselves. The association with gay men, though, was certainly striking, and early on, many called the mystery disease GRID, for gay-related immune deficiency. Lots of people would talk about it as the gay cancer or gay plague. But the disease wasn't limited to gay men. It was turning up in hemophiliacs, people whose blood doesn't clot properly and are treated with clotting factors taken from other people's blood. Doctors were also seeing cases in IV drug users, women, infants, and heterosexual men. In particular, 20 recent immigrants from Haiti had gotten sick, and none said they were gay. Those clues were important because they told scientists the disease, which had finally been given the name AIDS, was probably infectious and probably transmitted by blood. There were other diseases that spread in similar ways, like hepatitis B. So, in March of 1983, the CDC issued a warning that doctors needed to be careful about blood transfusions, and that the disease seemed to spread through both gay and straight sex. Debates about the specifics, including whether it could spread through saliva, would happen later. But what was the infectious agent? The race was on for scientists to figure out what was causing the disease. French molecular biologist Luc Montagnier suspected a virus because at the time, the blood products hemophiliacs used were filtered for things like bacteria and fungi, but viruses were too small to catch. So, along with his colleague, Françoise Boré-Sinoussi, he searched cells taken from AIDS patients and found a retrovirus. Around the same time, Robert Gallo at the NIH in the US also identified a retrovirus in samples from AIDS patients. Both groups published their work in May of 1983, and shortly afterward, another team found yet another retrovirus. All the viruses had been given different names, and at first, it wasn't totally obvious that they were the same thing, but they were, and in 1986, the cause of AIDS had been given an official name: HIV. So, HIV was the problem, but where had it come from? And why had the epidemic struck now, in the decade of big hair, and Michael Jackson. While some researchers were scrambling to identify whatever it was that made AIDS infectious, others noticed that macaque monkeys also seemed to suffer from an AIDS-like disease. One group decided to take some blood samples from these sick monkeys, and in 1985 they found a virus that was similar to HIV. It was eventually called SIV, for simian immunodeficiency virus. Researchers started to think that HIV might have come from our primate relatives, jumping the species barrier. After a lot of work, they figured out that the virus behind the epidemic was very similar to the chimpanzee version of SIV, and they were the ones who had passed it to us. But how, exactly? There's no real way to put this delicately, but most scientists agree that the reason why SIV made the leap into humans, what's called a spillover, is because we had a taste for bushmeat, or wild game. In this case, monkeys and chimps. This is known as the cut hunter hypothesis. In the course of butchering a chimpanzee, some SIV-infected chimp blood enters a small cut on the hunter's hand, or a bit of blood splatters in their mouth. The virus is close enough to human biology to infect the hunter, and over time, if the hunter passes the virus along to enough people, it evolves into the HIV we know today. Spillovers like these happen many times. We can tell because the virus mutates quickly, and by looking at the genetic differences, we can identify multiple lineages of the virus, each one corresponding to a spillover. We've traced the current epidemic to just one of these, called M for Maine. By analyzing chimpanzee pee and poop, researchers think the chimps who passed that version of the virus to us lived in southwestern Cameroon in the forests near the Congo. And based on the oldest blood samples we can find that we now know have HIV in them, which are from 1959 and 1960, scientists estimate that HIV-1 first infected humans around 1908. If that seems like a long time ago, well, it takes a while for a virus to take off. By the 1920s, it's thought that the virus traveled downriver, in a person, of course, to the burgeoning city of Kinshasa, then known as the Belgian colonial city of Leopoldville. There weren't many women around other than prostitutes, so experts think HIV spread that way, and possibly through injectable drugs the colonists used to treat some tropical and venereal diseases. This was before disposable syringes, and nurses were trying to treat lots of people with just a few of them, so the syringes may have only been rinsed with alcohol before being used on the next patient. So the very methods meant to stop the spread of disease may have actually been encouraging it. With time, infected people in Kinshasa left to go to other places, and they did the unavoidable. They brought the virus with them. Because the virus mutates so quickly, we can group the viruses into 9 different subtypes and get a sense of how HIV traveled around the world from Central Africa. Several subtypes spread to other parts of Africa. Subtype C went south and then landed in India. Subtype B went to Haiti, and then, through several quirks of history, came to the US. First, in 1960, when the Belgians left the Congo, French-speaking Haitians started to arrive in the Congo to work as doctors, lawyers, and other professionals. But with the creation of Zaire in 1965, the immigrants felt unwelcome, so they went back to Haiti, bringing HIV with them. There, HIV expanded especially quickly, possibly because of a plasmapheresis center where people could get paid to donate their blood plasma. The center used a machine that mixed the blood of different donors, allowing viruses to transfer. By 1982, nearly 8% of a group of young mothers in a Port-au-Prince slum were HIV positive, an astoundingly high number. HIV is thought to have entered the US around 1969 with just one infected person or unit of plasma from Haiti. It took about a decade for anyone to notice, but by then it was too late. The epidemic had begun, and HIV was not only in the Americas, but Europe and Asia too.
As it was described in this video, the closest relatives of HIV-1, as determined by DNA sequence analysis, are simian immunodeficiency viruses, SIVs, that infect chimpanzees and gorillas in the West Central Africa. And in most cases, they don't cause severe disease in their hosts. The SIV, that is most closely related to HIV-1, is carried by chimpanzees, and the HIV-1 strain M, which is responsible for the global human AIDS pandemic, is most likely to have been derived from a chimpanzee in the southeast Cameroon early in the 20th century. However, HIV-2, which is responsible for some cases of AIDS in West Africa, is a distinct virus, most closely related to SIV, whose natural host is the sooty mangabe, not chimpanzees, an old world monkey found in West Africa. The different strains of HIV-2 represent separate zoonotic acquisitions of simian virus from these monkeys. What about malaria? There are many different species of Plasmodium that infect wild non-human apes, yet only 2 Falciparum and vivax have successfully crossed over to infect and transmit between humans. Falciparum is the most closely related to Plasmodium that infects western gorillas in the wild, although it has been also infecting chimpanzees, bonobos in captivity. The origin of vivax, which causes milder malaria in humans, is less clear. Vivax-related parasites infect other ape species, yet Plasmodium, most closely related to vivax, infects macaques in Southeast Asia, here, suggesting that these might be the origin of the zoonosis. The transfer of pathogens is not one way, because there are examples of human diseases transferring to other primates. This process is called anthropozoonosis, particularly in situations where there is a close contact between species. Human-derived falciparum can infect captive bonobos and chimpanzees. Chimpanzees can also suffer from the respiratory disease caused by human respiratory syncytial virus, or HRSV, very common virus in the humans, and the human's metapneumovirus, HMPV, both of which are a major cause of respiratory disease in humans. And phylogenetic analysis of viral DNA sequences confirms that the viruses infecting chimpanzees originated from a human virus within the previous 10 years, only in 10 years. These and other possible cases of anthropozoonosis has occurred in habituated non-human ape populations where free-living chimpanzees or gorillas have close contact with humans because of tourism or research. The transfer of the disease between apes and humans is possible because we are closely related species. But how closely are we indeed related? Let's find it out in the next part.